This video is about rabies and hantaviruses. Feel free to download your Microbiology It workbook. The link is in the description box. Without further ado, let's microbiology it. Rabies is one of those infections that reminds us how closely human health is tied to wildlife. It's caused by a rhabdovirus, a single stranded negative sense RNA virus that specifically targets the central nervous system of warm blooded animals. Most human cases start with a bite from an infected animal, such as raccoons, foxes, skunks, coyotes, and especially bats, are the primary reservoirs. What makes rabies particularly challenging is its long and variable incubation period. In some humans, symptoms may not appear for many months. But once the virus reaches the brain, especially the thalamus and the hypothalamus, it begins replicating rapidly, leading to the classic symptoms of fever, agitation, dilated pupils, excessive salivation, and the well known hydrophobia caused by spasms in the throat muscles. Without immediate post exposure treatment, the infection almost always progresses to respiratory paralysis and death. What makes rabies so distinctive is how it's diagnosed and managed. Lab confirmation typically involves fluorescent antibody tests on brain tissue after death or identifying Negri bodies, viral inclusion bodies, in infected neurons. But in practice, public health decisions often rely on the behavior of the biting animal. A domestic dog or cat that bites without provocation is quarantined for 10 days, while wild or stray animals are tested right away. Thankfully, modern rabies prevention is extremely effective. Passive immunization with human rabies immunoglobulin at the wound site plus vaccination with inactivated rabies virus can stop the virus before it even reaches the nervous system. For people who work with animals, veterinarians, wildlife handlers, and lab researchers, pre exposure vaccination provides an additional protective layer. Hantavirus infections are completely different in transmission and behavior, yet they share the theme of humans interacting with animal reservoirs. Hantaviruses are enveloped, single stranded negative sense RNA viruses related to pathogens like Lassa and Ebola, but their reservoirs aren't wild carnivores, they're rodents. Infected mice, rats, voles, or lemmings shed the virus in urine, feces, and saliva. Humans typically become infected by inhaling aerosolized particles from rodent droppings, especially in enclosed spaces. In the Americas, this exposure can lead to pulmonary syndrome, a condition that starts with fever and muscle aches, but can rapidly progress in respiratory distress, shock, and cardiac complications. In parts of Europe and Asia, other hantaviruses cause hemorrhagic fever with Raynaud syndrome, which includes intense headaches, abdominal pain, bleeding disorders, and kidney dysfunction. Even though hantavirus infections can be severe, diagnosis and medical management are more supportive than virus directed. Immunoassays can detect antibodies against hantavirus, and RT-PCR can identify the viral genome in circulation, but there are no specific antivirals or vaccines. Care focuses on isolation, rest, rehydration, and maintaining organ function. Because prevention is the strongest tool, limiting contact with rodents and eliminating rodent habitats are the key control strategies. Keeping food tightly sealed, reducing clutter, and avoiding sweeping and vacuuming areas with rodent droppings, methods that can aerosolize the virus, are all simple but powerful steps.